Have Dr. José Yáñez, a professor at the University of Chile, presenting for us. Dr. Yáñez currently serves as the Dean and full professor of the Faculty of Veterinary and Animal Sciences at the University of Chile. He leads a research group specializing in aquaculture genetics and genomics, focusing on implementing new strategies to sustainably enhance fish farming production through genomic technologies in the Americas, Australia, and Europe. He's published over 100 peer-reviewed scientific papers in international journals, making significant contributions to the field of aquaculture genetics and genomics. So today, Dr. Yáñez will be presenting the next steps in aquaculture breeding beyond genomic selection. So I'll turn it over to you now, Dr. Yáñez.
Thank you, Gabriela. So first of all, I would like to thank this invitation to participate and show a general perspective of modern genetics in aquaculture breeding. Basically, there is some bias in this presentation in terms of that we have a strong focus on salmon aquaculture, but most of the developments that I'm going to show here are applicable to other species. So please let me know if you can hear me okay. Everything is okay, I assume. So why aquaculture? Why to focus on aquaculture? It's not news that we need to produce more food with limited land availability. So there is land and soil crisis, right? So there is increasing also demand of food due to the population growth, and the land is a scarce resource, and our planet is covered by water in the 70% of its surface. So why not to think into aquaculture or aquatic organism when we think about increasing food production for an ever-increasing demand. So there is another important aspect of related to aquaculture species. It has been shown that production of protein based on aquatic organism is highly efficient, at least more efficient than terrestrial animals. So you can see here something important. Feed conversion rate is almost one to one compared against chicken, pigs, and cattle. So it's a highly efficient type of production for animal, for protein for human consumption. Another important fact is that fisheries from cod are stagnant at this stage. It is a consensus that populations from the oceans, populations of fish from the oceans are diminishing, decreasing in terms of availability. So it's harder to increase production of protein from fish using fisheries by now. And aquaculture or farming of aquatic animals, it's increasing very rapidly since the mid of the 80s, when there was a very important development for aquaculture globally. Another important fact is that farm fish already surpassed the production of beef. Beef is a very important source of protein, but during 2013, the total production of beef was surpassed by farm fish. So you can see here also the exponential growth very rapidly of aquaculture around the globe. This figure is even more contrasting right now, and there is a lot of developments on technologies associated to aquaculture in basically two different modes, right? So there is a very important development on inland aquaculture for freshwater species, mainly fish and also crustaceans. Shrimp is also another important aquaculture species, but also on marine aquaculture. So here we have a picture of tilapia farm in Ecuador. It's an inland aquaculture modality. And here at the right hand side, we have a coastal salmon farm down here in Chiloé Island in Chile. So both types of aquaculture are increasing across the years. These are the main species. So we have carps as the number one, mainly produced in Asia, China, and other related countries. Then we have also miscellaneous freshwater fishes like tambaqui and pacu in Brazil, and also some others in Asia. Shrimp and prawns, tilapia, with an 8% of the total amount of, or the total volume produced. Salmonids, 6%, and fresh crustacean, 4%, and also some other seawater and pelagic fishes. So most of the aquaculture is based on this already developed species in terms of techniques and management. But there are also some other emerging species and different types of emerging species, mollusks, crustacean, and also fishes. When we go to see the top 10 producers, China is the main global producer of aquatic organism through aquaculture. Then there are some other Asian countries: Indonesia, India, Vietnam, Philippines, Bangladesh, Korea, and then the first European countries, Norway, and then Chile from the Americas, and Egypt. So this is very important because most of the aquaculture is concentrated in Asia, but there are some other key countries, key producing countries in Europe, Americas, and the Americas. So if we go to value, so China produce a lot of fish, but not very high value fish. Then you can see that there are other countries here that surpass or pass to different levels of this ranking due to that they produce a highly value aquaculture products. Like the main example here, and I'm going to talk about most of the presentation in this case is the case of Chile, in which our aquaculture is mainly based on salmon aquaculture, a product with high value, and this also generates the possibility to incorporate different technologies and different innovative approach, such as selective breeding. So just for you to know and to get familiar with the Chilean situation, so Chile is a very narrow and long country down here in South America. We basically have, if we have to divide the country in three parts, we have basically here up north, very dry desert with a strong mining activity. Then the Central Valley is mainly for production of fruits and grapes and wine. And then in the south is basically aquaculture. Aquaculture is the main activity for the southern part of Chile. So these are the key agribusiness activity in Chile. By far, salmon and trout is the main largest agribusiness activity generated in Chile. It's the second largest exporter activity below copper mining, which is basically our main income by export. So salmon and trout is very important for our small economy. Then we have cellulose, grapes, wood, apples, cherries, and wine. So basically fruits and also forestry companies based on agribusiness activities. Okay, so it's important to tell that salmon species are not native from South America. All of the species that we farm here, Atlantic salmon is not, it's exotic. Pacific salmon from North America is also exotic, and rainbow trout, it's also not non-native. So we base our production in 3 different exotic or non-native species: Atlantic salmon, Pacific salmon, and rainbow trout. And we started to farm by the middle of the 80s decade. Then the production increased very fast, and we had a very strong issue caused by ISA infectious salmon anemia virus that generated a huge major crisis here in the southern part of Chile. But then the industry recovered, and now we produce about 1 million tons a year. So we're the second largest salmon and trout producer in the world, just below Norway. In terms of genetic improvement, okay, all of us, we know best liner and best predictor, the BLAP methodology, which is, I would say, the gold standard for genetic improvement and selective breeding in terrestrial and also aquatic animal species. These are the mixed model equations. So we all know what every term in the equation means. I'm not going to go in detail on this approach. And then we also have to estimate variance components, including additive genetic variance and environmental variance, to estimate heritabilities, and then lambda to be included into the mixed model equations to solve the to estimate the fixed effects, and also to predict the random effects or the animal genetic effects. So something very important here is that the A matrix is built using pedigree information only. So this is the conventional, what we call conventional or ordinary BLAP, using only information from pedigree to reconstruct the covariance between animals, genetic covariance between animals. So something also very important in terms of aquatic organisms is that the genetic gain has been very impressive compared to terrestrial animals. And this is mainly explained by the high fertility rate and from the aquatic organism, and also the very recent events of domestication of this species. So as you can see here, by 2010, tilapia reached same amount of growth rate gain that chicken, that was one of the first species, that animal species that with very early breeding programs to improve growth rate for this particular species. So we reach genetic gains up to almost 20% of gain of genetic progress or rate of progress per generation. So that this is also a very important aspect of aquatic organism selective breeding. Salmon already surpass swine, and shrimp is the last species in with breeding programs established using BLAP methodology, but still the gain is very fast. So generation interval also it's also important for some of these species. For instance, in shrimp, you can get 1 generation every 6 months, depending on the management conditions. but it's a very, very fast species to demonstrate the value of genetics and breeding to the producers. Same for tilapia. Intergeneration interval is 1 year. This is not the situation in salmon. Atlantic salmon has a 3 to 4 years generation interval, but in general, fecundity and also high diversity, genetic diversity, it's very important to reach this kind of genetic gains. So these are a couple of examples. These are the genetic trends for population, coho salmon population from Chile, in which a breeding program was established in 1998 to improve harvest weight and also to measure and improve the flesh color, because it's also an important trait for the breeding objective to be included in the breeding objective. And you can see here that the genetic trends represented almost a 10% of genetic gain per generation. That means about 250 and 300 grams for a fish that grows to 2.5 kilos or 3 kilos. So it's very rapid. These generations, this population already have more than 10 generations. So that means that the current fish almost or even surpass the double growth rate from the founders. In tilapia, it's even more extreme. So you can see that this is the response to selection for gift tilapia, genetic improvement of farm tilapia in Asia. I'm sorry, these slides are in Spanish. But in general, what you can see here is that almost a 20% of genetic gain as a response to selection per generation was obtained in this gift breeding program. So every generation here is a year. So in almost 5 years, they duplicated the growth rate almost for this population that is already distributed around the globe. In terms of genomic technologies, we recently published a review paper together with some people from Norway, Brazil, China, and Sweden, in which we generated a very comprehensive article describing all of the genome-wide association studies and also genomic selection publications. We put together all this information with a critical discussion in terms of aquaculture species. So if some of the things that I'm going to talk in this presentation and the details are in this article. In terms of division, so there is this paper from Sewell Wright, Genetic Principles Governing the Rate of Progress of Livestock Breeding, published in the Journal of Animal Science in 1939. So as a starting point, suppose that we were given a reasonable complete map of all of the chromosomes showing the location of all important genes affecting the character in question, as well as of convenient marker genes. What could we do with it? We know now what we can achieve with different levels of resolution of molecular markers or genomic information to improve or increase the rate of genetic progress in livestock and aquaculture species. So we also know that cost of genomics have decreased very rapidly across the years. So now we can even sequence a genome. For instance, there are some techniques like low-pass sequencing or skim sequencing, in which you can sequence an animal in 1x coverage or half X coverage for $30 to $40. So this is very important in terms of the developments that we can reach for this in animal genetic improvement. Maybe all of us are familiar with the SNP technology. So in aquatic animals operates very similar to terrestrial animals. You take biological samples from fin clips. Generally, you purify DNA. You can sequence this DNA to discover SNPs, and then you can also use SNP genotyping platforms to do high throughput DNA genotyping by using Illumina technologies or Affymetrix, now Thermo Fisher technologies. There is something interesting here regarding that most of the terrestrial animals, they have their SNP arrays or their SNP chips developed using Illumina technologies, but for aquatic animals, most of the something it's just something that I found it interesting. Maybe it's related to the negotiation between every developer and these companies. We developed, when we started, when we wanted to implement genomic selection for Atlantic salmon, we didn't have the technology in place to do it. So the first thing we had to do early in 2011, we started with a project, a national project here in Chile, to develop an Atlantic salmon SNP chip. So we participated, we led this development of Atlantic salmon SNP array together with some collaborators from Université Laval, some other collaborators from Scotland, and also from the industry. Genus PAC, Pig Improvement Company, was one of the companies that helped us to develop this SNP array. We had two versions, one of 200K SNPs, and also another one with 50K SNPs for routine genetic evaluation or genomic evaluations analysis. Then we developed this SNP array. I would say is one of the few SNP arrays with Illumina technology in aquatic animals for tilapia, including diversity from South America and also from different parts of Asia, as including some populations from South America that has Asian origin. And then we also participated in the coho salmon genome project together with a Canadian consortium, in which we also developed a 200K SNP array and also 70K SNP array for coho salmon, Pacific salmon. We also participated in the development of the Eastern oyster SNP array. We participated in the validation of the shrimp SNP array, and also with a Brazilian group, we sequenced some animals and also developed SNP array 30K for pacu and 30K for tambaqui, two of the most important native species for aquaculture in Brazil. So we have basically very important set of tools to implement genomic selection schemes in the most important species, aquaculture species farm in the Americas. So what we can do with the SNP chips is basically at the beginning, what we always do is to run genome-wide association studies, or GWAS, to find some QTLs or genomic regions that are explaining an important part of the genetic variance for different traits. As you can see here, we have found some interesting QTLs for different traits. In Atlantic salmon, for instance, this is a flesh color QTL, so an important region in chromosome 14 that is explaining astaxanthin pigment retention in Atlantic salmon. We found a BCO gene, beta-carotene oxidase gene, that is involved in this QTL. So this is an important development to implement marker-assisted selection for increasing the rate of astaxanthin retention or pigment retention in salmon flesh. Then we also validated QTL for IPN resistance. IPNV is virus, infectious pancreatic necrosis virus in Atlantic salmon, and there is an important QTL in chromosome 26 that has been successfully used in Norwegian and also Chilean industry to control this disease. And then we also found an important region in chromosome 21 for bacterial resistance, SRS, septicemial or septicemial rickettsial syndrome resistance in Pacific salmon in Chile. So, but as you know, not these are very particular cases. Not always we have this kind of important regions containing a high percentage or a high proportion of the genetic variants for a particular trait. But in the case of the QTL for IPN, there are some very interesting results in the field. So this QTL has been used to improve the resistance against this virus in Atlantic salmon by the use of marker-assisted selection, using haplotype-based selection, or even marker-based selection, in which also there is dominance from the favorable alleles. For instance, here we have the homozygote for the good alleles, and you can see here the mortality, dead or alive, and the frequency of the or the genotype frequency, and these are the combination heterozygote combination, and then the homozygote for the bad alleles in the QTL. So this is a dominant QTL in which survivors are, or the survival rate is even increased when we have only one dose, one copy of the good allele in the genotype for this particular marker. So these are official data or results from the Norwegian Veterinary Institute, in which we can see here the decrease in mortalities either for seawater or for freshwater across the years. So the QTLs started to be commercialized, or the X with the incorporation of this technology started to be commercialized during 2010. So you can see here, or 2009, I'm sorry. So you can see here how the mortality started to decrease when the commercialization of eggs with this QTL started to happen. The same for the number of outbreaks here. So as you can see here, this is the IPN QTL-X. That means the X which incorporated the QTL for IPN resistance in the X or in the technology, the genetic technology. And then you can see here the outbreaks, how they started to decrease when the X were increasing in terms of commercialization. So this is a very successful example of the application of a QTL for disease resistance in salmon aquaculture. But then not all of the traits are explained, or the variation of these traits are explained by important QTLs. So genomic selection is also another option to increase the rate of genetic gain or genetic progress through the increase of accuracy of selection by using genomic information plus pedigree and phenotypes. So this is the way that genomic selection operates in aquaculture. We generally produce different families, different sets of families, from 100 to 800 or even 1,000 families. Then you have the selection candidates, or the candidate population that is pristine, and you only tag these animals and take samples to get genotypes. And the reference populations, the collaterals, go for phenotyping activities like disease challenges, genotype and environment interaction, and also processing plant assessment of traits like flesh color, feed yield, or carcass quality. So these animals are phenotyped techniques, and then you get the phenotypes or genotypes from these animals to train the models, genomic prediction models, and also to build the genomic relationship matrix using dense SNP panels. Then with the genomic prediction equations, you estimate the breeding values, or the GEBVs, genomic breeding values, for the selective or selection candidates to make selection decisions at this stage. So it's basically similar to what we see in other terrestrial species. We have candidates for selection that are not phenotyped, only genotyped, and then we have reference population that is or training population that is that is composed of animals with phenotypes and genotypes. And also you can incorporate pedigree information. In Atlantic salmon, at least the situation in Chile is that viral diseases, the most important viral disease is IPN, infectious pancreatic necrosis. You can see that most of the genetic variance is explained by a QTL in chromosome 26. This is almost 80% of the genetic variance explained by this QTL. So it's a very big QTL. Then we have sea lice that is explained, the variance is explained by different regions in the genome, including almost all of the chromosomes with no QTLs, polygenic architecture for this trait. The same is noted for SRS, that means bacterial resistance, with some small, very small QTLs distributed across different chromosomes, but all of the genetic variance explained is distributed along the whole genome. So when we talk about genomic selection in practice, I don't know your experience, but in general terms for aquaculture, single-step GBLAP is the gold standard methodology, given that it's easy to implement, it's very efficient from the computational point of view, and also you can include animals that are genotyped, that have genotype information, and also pedigree, and also animals with pedigree only. So it's a very interesting approach that describes this H matrix that is a blend between the pedigree relationship matrix and the genomic relationship matrix. So here are some examples that we develop as a proof of concept for bacterial resistance, SRS resistance in the three main species for Chilean aquaculture: Atlantic salmon, rainbow trout, and coho salmon. We tested different methodologies for two trait definitions, date of death, and also binary survival for the resistance to this bacterial disease. And we compared genomic models using different approaches, frequentist and also Bayesian approaches against ordinary pedigree lab, here in this case in orange, and then all of the genomic approaches are plotted using these light blue bars. So we use GBLAP, Bayesian lasso, Bayes-C, and also single-step GBLAP to compare different genomic selection models to pedigree BLAP, and we reach a different percentage of increase of accuracy in the prediction of breeding values when using genomic information. So then for another important trait, sea lice, which is globally, it's a global threat for salmon. There are some empirical studies. Here we have worked with a company. They have two experimental lines. So these are results from three generations of genomic selection, for selection for high resistant and low resistant lines, and also a control line, not selected line. That means when I'm talking about resistance, I'm talking about of lice count or lice load, lice burden in the animals. So we define resistance as lice load, and then we notice that there is a great response to selection in a very short time. So in 3 generations, we reach a cumulative 73% of genetic gain. That means 20% of lice count reduction per generation. So genomic selection is working in aquaculture, and it's also improving what we have done using only pedigree only. But there are some new challenges, and genomic selection is here, so it's not futuristic. It's implemented in routine genetic evaluations in different aquaculture species, including salmon, trout, tilapia, and also shrimp. But there are some new challenges, and I'm talking about here on what we're focused now. So genetic gain is controlled by selection intensity, or is influenced by selection intensity, by accuracy of selection, or accuracy of predictions, and also genetic variance, genetic variation of the population, and and also influenced by generation interval. So in terms of accuracy, there is this paper of simulation from Miguel Pérez Enciso from Spain, in which he simulated a trait with different 1,000 QTNs, for instance. That means quantitative trait nucleotides, so causative mutations. And when we use pedigree, that means BLAP. The accuracy has his distribution in the simulations. The main accuracy is generally a little bit higher than 0.5. Then when we move to 70K SNP arrays, we move to 0.7 accuracy in general. And when we use whole genome sequences, contrary to what we expected, or what was expected, the accuracy didn't increase too much from lower density SNP panel, 70K to whole genome sequences. That means a jump between a leap between in the order on the tens of thousands SNPs to the order of millions of SNPs using whole genome sequences. So what Miguel proposed here is that when you use the actual QTNs, if this trait is controlled by 1,000 QTLs, and you use those 1,001K QTNs, you can maximize accuracy of selection if you use only this information from QTNs. So we are focused now on discovering these QTNs for different important traits, but this is not a simple task, as you can imagine. So one approach is to use imputation to decrease the cost in terms of sequence efforts. So you can predict missing genotypes using information from reference populations, and then you can impute from low density SNP panels to cheaper high density ones, including whole genome sequences. We actually use software facilitated by Gabor here in this study. This is a study carried out by Baltasar Fernandez. He took 30,000 fish, more than 30,000 fish, most of them genotyped with 50K SNP arrays, 10K fish with 50K, 1.5K SNPs, animals, genotyped with 200K SNP, and 1.3 animals genotyped with 60K SNP array. Then we have almost 300 animals with whole genome sequences. That means 6.2 million of SNPs. So we did a sequential imputation from 50K to 200K, then from 200K to 60K, and then from 60K to whole genome sequences, reaching a total of 2.5 million SNPs to be used in meta-analysis for disease-resistant traits. In order to refine and also to increase the statistical power to identify genetic variants involving the traits. So we recently published a couple of studies, one of them for sea lice. We did a meta-analysis of genome-wide association studies for sea lice load. In Atlantic salmon, we discovered different QTLs, including different potentially important genes. None of them has been characterized from the functional perspective, but still we have several important candidates to be involved in sea lice burden in Atlantic salmon. Then we also did the same for SRS, Piscirogetes salmonis, which is the bacteria that affect Atlantic salmon in Chile. We also refined some QTLs. And then something important from this analysis that we use multi-population analysis in order to increase the statistical power and also the accuracy of detection of QTLs. Tens of thousands of fish were used, so we increased also the number or the sample size for the studies, trying to have more power, and then we also tested prioritization of genetic variants in order to know if we could increase the accuracy of breeding value predictions. And also we have found some interesting candidates for gene editing, and we are testing gene editing in vitro using cell cultures in order to narrow down the list of important targets to be tested in vivo in this for these traits. For this one, for this trait, it's very tricky because it's a highly polygenic trait. But then for SRS, Piscirogetes salmonis, we found some interesting genes. But anyway, they don't explain a high proportion of the genetic variants. So they're important from the functional side, but still there is a lot of work to do in terms of discoveries of QTNs for these particular traits. So in terms of the prioritization of SNPs for genomic selection, we did imputation to whole genome sequencing. So we used about 2.5 million imputed genotypes. We trained the data for sea lice count in 5 different populations: 2012, 2013, 2014, 2016, and 2017. And then we used this training population to validate the accuracy of EBVs, GEBVs, genomic estimated breeding values, with a population in which we also have genotypes and phenotypes, and the phenotypes were masked in order to estimate the correlation between the predicted phenotype against the true phenotype, the actual phenotype. So we generated different sets of SNPs in order to test accuracy. So we had 70K SNP panels, so simulating they were randomly distributed, and then we used also 70K, 10K, and 6K prioritize SNPs using based on the variance explained in descending order. What we can do here is that using 70K SNPs, prioritize SNPs in light green, we surpassed in two different challenges, in two different phenotyping events, the accuracy of prediction performed using 70K panel with SNPs randomly distributed across the genome. But when we reduced to 10K and 6K, the accuracy of selection, or the accuracy, I'm sorry, for breeding values estimation decreased a bit. So this still has to be validated, but we believe that if we can prioritize SNPs, we should see an increase in accuracy of breeding values prediction, according to what was simulated by Miguel Perez Enciso in that paper from 2015. Then there are some challenges related to the integration with other omics. We are working on using computer vision and also some machine learning algorithms to decompose the flesh color phenotype into RGB spectrum in order to automatize the phenotyping of color in the flesh in Atlantic salmon and also rainbow trout. So this is be done by Rodrigo Marin. Oh, I'm sorry, I cannot run this video. There's supposed to be a video here. I can show you later. It's also some algorithms that are being used to estimate fillet yield from a picture that is taken in the processing plant for different individuals of pacu and tambaqui. So it's native from Brazil, fish native from Brazil. And then the challenge is to recognize individually and also taking measures individually from the fish using computer vision or other technologies, capturing videos and transforming that videos in longitudinal data across the whole period of sea farming of these animals. Then another direction that we're taking is to incorporate microbiome information into genomic prediction, specifically for sea lice resistant in Atlantic salmon, using skin samples, skin microbiome samples. So there is a paper here, very recent, from Lucas Venegas. We have this work in collaboration with Université Laval and also Benchmark Genetics, in which we are trying to identify some parameters, let's say microbiability, which is the variance explained by the microbiome in terms of the phenotype. And also there are some other interactions between the genetics of the host and the phenotype that are also widely known to be controlled by heritability, and also the genetics of the host and the interaction with microbiome, the influence of the host in the microbiome, and also the influence of the microbiome on the phenotype through the microbiability. There is another term that has to do with the interaction of host genetics and microbiome genetics that is called the holobiability, which is related to the variance explained by the interaction between the host and the microbiome. So this is very preliminary. So Lucas already has his data, and what we can say that for this particular trait, for for sea lice, response to sea lice or resistance to sea lice, microbiability is even higher than heritability. So there is a strong importance on microbiability or in the variance of the microbiome that also can be controlled by the genetics of the host in terms of the sea lice count in Atlantic salmon. So we have an hypothesis here, an hypothesis that is related to the presence of or the abundance of certain microbes or bacterial groups or genus that generate organic volatile compounds that can be attracting more lice to the skin surface in Atlantic salmon. But this is a spoiler of Lucas' work. He's about to publish this study, which I found very, very interesting. And this is the final slide. So we started here in the, or maybe if you don't remember, PCR was developed during the 80s and then was distributed or implemented across labs in different parts of the globe during the decades of the 90s. So PCR was a technology trigger. This is the Gardner hype cycle. That is a curve that explains very good the implementation of new innovative technology um, in the field. So we have the PCR as a technology trigger. So we have, by that time, we have low density SNP markers, QTL using linkage analysis, using families, some QTLs that were presented or were shown in some families, but they were not segregating in the same linkage phase in other families. So they were very hard to implement in practice. There was some power of discoveries with very sparse markers across the different chromosomes by using linkage or family recombination across families. The main markers used were microsatellites. So after knowing that microsatellites and QTLs were not applicable to genetic to breeding, to animal breeding, most of them were not applicable because they were segregated in only some families, or the linkage phase was not always the same for different families. And after this peak of inflated expectations, there was a truth of disillusionment. And then with the development of high density SNP panels and genomic selection, theoretical approaches that were describing this methodology. For instance, in 2001, the paper from Ben Hayes, Mike Goddard, and Theo Mewisen describing the use of SNPs to implement genomic selection was critical to start new developments. And I would say for animal breeding, the plateau of productivity was reached during the implementation of single-step genomic selection here for the actual routing implementation of genomic information into genetic evaluation schemes for different terrestrial and now aquaculture species. So single nucleotide polymorphism, we have a better picture of what we have in genetic terms of the different animals, different species. This is an Atlantic salmon. Then now in 2020, we're working with ultra high density, trying to find QTNs using whole genome sequences. So we have a better picture, more resolution on the genomic information, but still there is a novel technology, not that novel, but still very promising technology, gene editing. Everybody's talking about this, but this technology trigger, I think we're starting, and this is a very personal vision, that we are starting a new Gardner hype cycle in which we have a peak, or we're reaching a peak of inflated expectations, and then we're going to live through this disillusionment, and we need some enlightenment or some new insights in order to know and to reach a plateau of productivity of gene editing based on QTNs that are really hard to discover. Yet I know functional information from eQTLs and allelic specific expression and global gene expression, and also in vitro studies using different cell cultures to test QTLs and QTNs will be very useful, but still we are waiting in order to know how this technology will be deployed, especially for polygenic traits. For monogenic traits, it's true that we already have some developments already there. For instance, the Pierce PIC developed by PIC, Pig Improvement Company, and also the Roslin Institute and other groups is there. The FDA, I'm sorry, the FDA is waiting for its approval. It's expected to be ready for 2026, but still, even though for monogenic traits is kind of plain from that perspective, for polygenic traits there is still a lot of work to do. So that's all. Many thanks for your attention, and I'm open to questions.
Thank you, Dr. Yáñez, for your presentation. I'll open it now. We have a few minutes, but I'll open it to any questions from anyone in the room or online. Bruno has a question, so.
Hello, Dr. José Manuel. First of all, congratulations for our great talk. I took a lot of inspiration from your talk. So my question is, to conduct a breeding program with aquaculture species, for example, what's the fry producers' participation? What's the producers' participation? What's the industry participation to conduct? Because I guess every producer buy the fries for a company. So what's the participation of each actor in this process?
Yeah. Thank you for the question, Bruno. This is very important because at least in Atlantic salmon there are mainly three big companies related to genetic products. So it's Benchmark is there, Aquachem from EW Group, and also Hendrix. But there are some other companies that are fully integrated. There are big companies, and when I talk about big companies, I'm talking about companies that produce more than 10,000 tons of fish a year, right? So they have their own breeding programs. We have, for instance, here in Chile, we have at least between 8 to 10 breeding programs. 3 of them come from the breeding companies, the 3 that I already mentioned, but there are some other breeding programs that are carried out by companies. So they are fully integrated. There are some companies that produce even about 100,000 tons per year. So they are very big. And there is also another important point related to the margin of the operation. Salmon and shrimp, they have a good margin. But for instance, if you go to Brazil for tilapia has grown a lot in Brazil, but it's still very segmented in terms of numbers of producers. They are not big producers. It's very rare to find producers in Brazil that they have they produce more than 10,000 tons a year for tilapia. And also tilapia have a very low margin, so not good money to invest in these developments. Genotyping is very expensive, and also managing a breeding program is also expensive. It's an investment, but still not all of the companies and not all of the species can afford breeding programs if there is not a coordinated effort.
Thank you very much.
You're welcome.
David, would you like to ask your question?
Sure. Yeah. Thank you very much for that great presentation. And there's only a couple fish professors at this department and a few across the university. I see Liz Bolding's online. That's good. So I look at the gut microbiome, and I was really interested in those last slides. It can be very difficult to kind of compare two highly variable things like genomics and the microbiome. how are you going to incorporate that and adjust for that huge variation between the two? And what are your plans for kind of linking those two together?
Yeah, there are different ways of modeling microbiome variation together with genomic information. So you can model only the most simple is to model microbiome without incorporating genomic information from the host. But then there are some other more complex modeling approaches in which you can use the interaction between both in what is called recursive models. They have been described in the literature, but still is challenging, and but results are very encouraging. And also, I would say modeling is still in development. There are different approaches, as I mentioned. We have tested some of them, but I would say from the practical point of view, there is an extra cost, right? We did this with 16x microbiome information, but from the cost perspective, it's also limiting, right? So,
yeah.
But from the modeling approach, there are some approaches out there. Some of them more simplistic, and some of them more complex. But as I mentioned, the results are very encouraging.
Yeah, great. And are you working with Professor Nicolas Derome from Laval?
Yeah,
great.
Nicolas
Yeah,
Derome.
I work with him as well. Yeah, that's great to hear.
Yeah. He's involved in the project. Yeah.
Awesome. Good. Thank you. Take care.
You, David.
All right, thank you. We have time for one more question. So Elizabeth, if you would like to ask.
Oh, you're muted.
You're muted, sorry.
Yeah, I was. All right. Thank you for your seminar. It was really good, and you really covered a lot of ground. And I just had one really small question. So you were talking about low coverage sequencing, and you were saying you were having good success at less than 1x coverage, and yet the people who work on wild populations, they're now saying you need about 5x to be really sure. Do you think you're getting away with using 1x or less because you are doing imputation from the parents? And if you are doing imputation from the parents, what coverage do you do the parents? That's what I'm.
Yeah, it really depends on the species. For instance, I would say it depends on the complexity of the genome. For instance, Atlantic salmon, they have a very complex genome, and it's hard to get good accuracies. When I talk about half x or 1x, I'm talking about tilapia, which is 1 gigabase size of genome, really simple compared to salmon or trout. And coverage for the reference is also important. So the accuracy that you get from imputation from low pass sequencing depends a lot on the size of the reference population. And I'm talking about more than 300 animals sequenced with 30x ideally or above.
Oh, 30x. So the parents and some of the reference, you would do 30x.
Yeah.
Okay.
Even if you don't have the parents, you can do genotyping. You can do sequencing on related population, and you can impute from three. What we have done is 300 animals sequence with 30x as a reference.
Right. So these would be 300 fish from the reference that you sequence at 30x, and you do better than using a high density SNP chip.
Not. I mean, it's not. We have not compared to the 50 actual 50K SNP array, but we have more markers available.
Yes.
In the order of near 1 million SNPs available with high accuracy. So let's say we have more information, right?
Yes,
but
so that's very helpful. Thank you very much.
Yeah.
That's
but still, Elizabeth, that doesn't mean having 1 million SNPs doesn't mean that you get more accuracy.
No.
Higher accuracy of estimation in breeding values.
Right.
Yep.
Thank you again, Dr. Yáñez, for presenting for us today.
Thank you.
This was a great discussion. The recording of this will be posted to YouTube, and we can send you those details later. But thanks again, everybody, for coming. We'll see you next week.
Thank you. Bye-bye.
Bye.